Okay, now we're on to part 2 of the Crossing the Membrane Lab. So today, if you look at the worksheet, you'll be able to fill in part 2 data table, the second data table, which will be putting a bunch of substances in order on that data table, and then you'll need to write a conclusion for all of your data. So after a night of sitting, the cell is still in the beaker, the iodine's still in the beaker, the starch has kind of settled to the bottom, so it's not making the entire column of water that black purplish color anymore. What we're going to be doing now, just to quickly go over the procedures, is student assistant 1 is going to remove the cell very carefully from the beaker. She's going to bring it over to a sink and give it a quick rinse off. So student assistant number 2, kind of, that'll work, that's fine. Just rinse it off for about a second or 2. Perfect, and then bring it back to the lab setup. And now she's going to cut open the cell using a pair of scissors and dump the contents of the cell into the small beaker. Good enough so that you can see what is inside of the cell. Okay, and if you look at the contents, you can see that yellowish color in the cell water. That indicates that one of your substances is still, or did make it through the cell for sure. So now, following the procedures, you now need to record data for starch. Did any starch get into the cell? Was starch in the beaker? And then you also are going to need to test for glucose. Student assistant number 1, does the iodine tell you if there's glucose in there?
No.
No, it did not, right? It only turned colors for the starch, not for the glucose. So to test for glucose, we're going to be using these little guys. These are called glucose test strips. Student assistant number 1 is going to pick up each test strip, and you're going to swirl the blue end of the test strip into the cell water, and you're going to swirl the other test strip into the beaker water, and let's swirl for about 30 seconds to a minute. Okay, that swirling gets lots of the juices onto that little square. Go ahead and pull that strip out. Just set it next to the cell water right here so you know which one goes where, and then do the same for the other. That should also be enough time, so take that out and set it next to the beaker water. So in a moment or two, we're going to be comparing the colors on those test strips to what shows up on the bottle. Before we do that, so take a look at the data tables. So right now, your big goal first is to write down where the substances are. And the idea is, did the substances leak through the cell or not? Most of them should still be in the beaker, but did they get through the cell? Okay, so you're gonna have to use the fact that this one's just yellowish in color, and then we're gonna look at these test strips to figure that out. Then to the second data table, we're going to be putting in order these different substances on this data table, going from largest to smallest. This is going to require you to use your critical thinking skills to figure out which order they have to go in based on if things got through the cell or not. 2 of them are already given to you. Okay, student assistant number 1, pick up the 2 test strips, make sure you know which is which, and you're going to check by holding them up to the bottle here to see if they have glucose or not. So which one do they compare most to? Probably in between the 2? Yeah, so what do you think? Is there glucose in the beaker? Is there glucose in the beaker then, based on that?
Yes.
Yes. Is there glucose in the cell based on that?
Yes.
Yeah. Okay, so there's glucose in both. So that tells you that glucose was in both places. You can fill that in on your data table. Okay. Okay, that's the end of the lab. We're gonna dump out all the materials and throw out the test strips. You can now fill in your second data table and write a conclusion. Done. Go ahead and stop.